so that's for the calcium regulation, right? Now let's move on to the detailed actin-myosin interaction, okay? So we said muscle contracts because actin interact with myosin. All right, these two proteins interact together. So this is actin filament, okay, actin filament. This is myosin head, right? On this figure, we see thin filament, which is actin filament here, and myosin extended from the thick filament on top. And here's one of the myosin head. Okay, so when myosin binds ATP, it has low affinity for actin. So at this stage, myosin does not interact with actin. It doesn't bind. It just walk around actin, try to find a good place. All right? And then it hydrolyzes ATP to ADP and phosphate. Okay? At this stage, the affinity for actin is still low. Right? So these two are the low affinity state. And then phosphate is released. During phosphate release, there's a conformational change of myosin head. Right? So also it changes the binding affinity to actin to become high affinity. So when myosin finds a good place on actin to bind, it releases phosphate, and then it's strongly bound to actin, and also a conformational change happens, right? Okay, conformational change. So at this time, myosin binds actin strongly. It holds actin, okay? And after phosphate release, ADP is then released in the next step. This myosin still has high affinity for actin. Okay, so it holds actin strongly. Right, this is the place where it generates force. Okay, it grabs actin strongly, so that's the force. We call this cross bridges also. Cross bridge. Okay. Cross bridge. Okay. And when the ADP is released from myosin, then myosin has high affinity for actin, for ATP. So when ADP release, the next ATP molecule binds to myosin. Okay? And when it binds to myosin, myosin has low affinity for actin. So it releases actin. Okay, and back to the step one. So let's go through this cycle again. When myosin binds ATP, it has low affinity for actin. It hasn't bind to it. And then ATP is hydrolyzed to ADP and phosphate. It still doesn't bind. It just walk around. It just at the nearby place of actin, but it doesn't hold. Right, then phosphate is released. During phosphate release, it induces a conformational change of myosin. At the same time, it binds actin strongly. So it pulls the actin filament. Right, this is the sliding. So we call it power stroke, the sliding filament. And then after that, ADP is released. While ADP is released from myosin, myosin has high affinity for ATP. So this comes out from myosin and ATP binds to myosin. When ATP binds to myosin, it releases actin from myosin because the affinity for actin decreases. Okay? All right, so we have a video for that.
Muscle myosin is a dimer with two identical motor heads that act independently.
Okay, actin filament.
The head has a catalytic core and an attached lever arm. A coiled coil rod ties the two heads together and tethers them to the thick filament seen on
Okay,
top.
thick filament.
The helical actin filament is shown at the bottom.
This is actin.
In the beginning of the movie, the myosin heads contain bound ADP and phosphate and have weak affinity for actin.
Okay, so it walks around, all right? Try to find a good place to bind.
Is released.
All right, phosphate release causing a,
The binding of the myosin head to
then it's strongly bound.
also triggers the force-generating power stroke that moves the actin filament. ADP then dissociates, and
Again,
ATP
ATP comes,
finds the
right?
binding site, causing the myosin head to detach
Okay.
from the actin filament.
Because the affinity for actin decrease.
ATP is hydrolyzed, which recocks the lever arm back to its pre-stroke state. Thus, like a spring, the arm stores the energy released by ATP hydrolysis, and the cycle can repeat. The actin filament does not slide back after being released by the motor head, because there are many other myosin molecules also attached to it, holding it under tension.
Okay, that's the contractin actomyosin cycle. All right, so the cycle begins. All right. So I asked you to read those terms, right? One of them is rigor mortis. What does that mean? When you say corpse, dead body, right? Rigor mortis refers to 僵尸，尸体的僵硬，僵硬的尸体。It means when animal dies, their muscle becomes stiff, right? So what happens during the muscle stiff is the protein inside muscle, right? So which step is rigor mortis?
ATP. Loss of ATP.
Loss of ATP here?
Yeah.
Here. All right, so, yeah, actually. But why? Rigor mortis doesn't happen immediately when animals die. Right? When we say you died, what happens? Because
Enhanced respiratory.
usually we say if you stop breathing and your heart stop pumping, then we say, or you're dead. Right? But in your tissue, in your cells, it's still alive. Your cells still alive. Still has some oxygen in the blood vessel. So your cell is still functioning. Muscles still can contract. Right? But after a couple hours, because you stop breathing, there's no more oxygen supplied to your cells. So, let's see. So no more oxygen and no more ATP generated, right? Without ATP, the actin and myosin were trapped here, right? So this happens after a couple hours when the cells, muscle cells, depleted, used up their ATP. So they go through contraction, but no more ATP can bind to the myosin. So it gets stuck in this state. Therefore, they cannot separate. Myosin cannot separate from actin. It's stiff. That's called rigor mortis. But after a couple hours, again, this is no longer happen. Then what happens?
Muscle is be degraded.
Yes. Because the cell membrane started to degrade, right? The enzyme stored in the cells were released, and the proteins are degraded. Okay? So that's completely cell death. Okay, because there's no intact myosin, no intact actin. They're degraded. Okay, so it's no more stiffness. All right, so the time when we examine the condition of the dead body, right? If it's stiff or if it's not stiff, okay, we can use that in the forensic investigation, right? We can estimate when the murder happened, like 12 hours ago or 2 days ago, okay? All right, so that's conclude our lecture today. We talked about regulation of muscle contraction, okay? How calcium induce interaction between myosin and actin, okay? And we look at the detail of actin-myosin interaction, the cross-bridge cycles. Okay, any questions? Are you following me so far? All right, not too bad. Okay, so let's do a little bit review. In this whole topic, Unit 7, we talked about excitation, contraction, coupling, EC coupling, okay? In skeletal muscle, it's regulated by calcium binding to the thin filament. So this is called thin filament regulation, specific for skeletal muscle, okay? And the cross-bridge cyclic interaction. And we have mentioned different scenarios like malignant hyperthermia, rigor mortis, okay? And yesterday we talked about muscular dystrophy. It's not functioning properly, okay? Or muscular hypertrophy. If you work out in the gym, your muscle gets bigger. Let's see. This is before the training. That's the regular muscle mass, okay? If you work out a lot, do a lot of exercise, your muscle get bigger. That's hypertrophy. Exercise-induced hypertrophy, okay? And dystrophy or atrophy means disease, usually disease causing a waste of muscle. It's called atrophy, dystrophy. Okay, let's see what's the answer. Which of the following protein is a component of thick filament? Okay, so in here, this is cytoplasmic protein. Okay, it's not part of the thick or thin filament. Okay, they're in the cytoplasma. These three are thin filament proteins. Okay, so myosin is the thick filament protein. And which of the following is a calcium binding protein? Troponin C, right? C is the indication. Okay, it binds calcium. And which of the term is best to describe him? Haha. Well, hypertrophy, right? Okay, which of the following is not a property of myosin? Okay, myosin is a hexamer. It has 6 polypeptide chains, which is not. Okay, so this is the answer. Myosin has ATP binding site. It binds actin, so it has a actin binding site. It is a ATPase. Okay, it binds ATP, and hydrolyzes it. And is self-assembly to form a bipolar filament, right? What's next? The signal pathway for EC coupling. Okay, so there are different events. Let's see. So it starts with the voltage sensitive calcium channel opens, right? So calcium entering the cell, and then it triggers another calcium activated calcium channel on SR membrane, right? And then calcium was released from the SR to the cytoplasma. And this calcium binds to troponin C and then induce a conformational change of tropomyosin to expose the myosin binding site on actin. Okay? All right, that's it, I think. Oh, there's another one. When actin and myosin interaction, which state will it be trapped when the cell runs out of ATP? Okay? It gets trapped here. So the myosin bound to actin strongly.
Or the ADP.
What about 这怎么了？为什么不能选这个？ This means myosin is not bound to actin. Okay, when I put this hyphen, it refers to they're bound together. In here, they're separate molecules. Okay, they're separated. It's not bound.
But ADP is not separate too.
没有 ADP 要先调出来。ADP 要先调出来。OK。
所以可以没有 ADP。
对。ADP 会调出来没有关系，可是，这时候他应该还抓着它。但是 ATP 调出来以后 ，myosin has high affinity for ATP， 所以通常是一调出来 ATP 马上就进来。那 ATP 没有进来，这个应该是 bound。所以我写错了吗？没有。是我的意思是说 ，is it not the state in that contraction cycle？ 那个 cycle 里面有这个 state 吗？没有，所以这个是我写错，我应该写这样，对不对？ Maybe this is also strongly bound, but in rigor state, this will be released, and because we run out of ATP, so ADP can be released. Maybe I should not put this. 这可能会比较困扰了哈，那就是可能题目出的不好，我不应该放这个，但是。 基本上在细胞里面的话，这个 ADP can be released. All right? Okay, that's it for today, and that's all for my lecture. 好，我们可以下课了。